So good morning, everyone. It's a great honor for me to start this meeting. And before starting, let me thank the organizer and Federico for the invitation. So as you heard, today I'm going to talk about the transcription factor MITF, simulation, senescence in melanocytes, but also in melanoma cells. So first, a few words on senescence. So senescence is referred to aging and age-related disease, such as cancer. This is a major tumor suppressor mechanisms that stops many cancers just as they start to grow. And this process is so important that Elizabeth Blackburn got the Nobel Prize in 2009 for her work on senescence. So senescence is said when cells exit the cell cycle and stop proliferating, and this proliferation arrest is not responsive to a mitogenic stimuli, which is different from quiescence. So senescence can be replicative or premature. Replicative senescence is associated with telomere shortening every time a cell divides. And this is what is called the Hayflick limit, reported by Hayflick and Morehead in 1961. Senescence can also be induced prematurely in response to stress. This is stress-induced senescence, or oncogenes. This is oncogene-induced senescence. And this senescence, this premature senescence, is telomere independent. So both types of senescence are morphologically indistinguishable and display similar markers, such as cell flattening and enlargements. They become positive for the senescence-associated beta-galactosidase activity. They display senescence-associated heterochromatin formation that can be detected by, for example, the heterochromatin protein 1. They display activation of the DNA damage response signaling pathway that can be detected by 53BP1 or activated form by phosphorylation of ATM and CHEK2. They can also display activation of tumor suppressor pathways such as P16 and P53. And finally, they can also produce an inflammatory secretum. So from melanocyte to melanoma, the process of senescence seems to be central in the melanocyte lineage. So in melanocyte, so you know that several pigmentary changes occurs during aging or photoaging because UV is a well-known skin aging factors. So here, this is a picture of senile lentigo, which is also called solar lentigo or H-spots. That is hyperpigmented spots that often develops on sun-damaged skins of the face or on the back of the hand, for example. The nevus is one of the most prominent examples of senescence in vivo. It is thought to be a form of melanocyte that enters the process of senescence following oncogenic activation, such as oncogenic BRAF V600E and NRAS Q61K or HER, which are the two main mutations identified in melanoma. And here, this is senescence-associated beta-galactosidase staining of nevus, and you can appreciate this typical blue nevi in this lesion, indicating that the nevus, are in a senescent state. And finally, several mutations, whether at the somatic or germline, in melanoma are related to senescence or the control of cell proliferation, which is CDKN2A, which encodes for the tumor suppressor P16, the retinoblastoma protein, or the telomere maintenance genes, POT1, HCD, TER2IP, and TERT. Another factor that is important in the melanocyte lineage is MITF, which stands for microphthalmia-associated transcription factor, and this is the master gene of melanocyte of homeostasis. So MITF is a basic loop helix leucine zipper factor, which binds as the dimers on specific sequences called E-box localized in the promoter of its target, in the locus of its target genes. And here, this is a mouse with MITF invalidation. And you can see that this mouse is white, and it is white not because it has non-functional melanocyte, like in albino, but because it has no melanocyte at all. And there is also some MITF mutation that can be detected in patients suffering from the Waardenburg syndrome type 2A. So MITF is also important for the differentiation and pigmentation process. And years ago, when I was PhD student, we showed that the cyclic AMP elevating agents, so here for scoline, this is cell pellet and in vials. And so you can see that cyclic AMP elevating agent stimulates melanin synthesis. And when we use a dominant negative form of MITF, the melanin synthesis is dramatically decreased in response to cyclic AMP elevating agents. And finally, MITF is also important for melanoma. And again, we show here, we identify the first pro-oncogenic mutation in MITF that predisposes carrier to melanoma, and we will talk about this mutation later on. So we don't know precisely how MITF controls these different biological functions, but a model proposed that the activity of MITF depends on its expression level, on its post-translational modification, among which simulation, and its cofactor bindings. And this creates a sort of barcode-like situation that channels MITF towards a subset of specific target genes and drives a different biological function. So today we will talk about simulation and senescence, as I previously told you. So because I show you that MITF is important for the development of the melanocytic lineage, and because MITF is also important for the proliferation of melanoma cells. Years ago, we thought that MITF was a good candidate to pursue as a target to control the proliferation of the melanocytic cells. And so for that, we used the approach of RNA interference to inhibit MITF expression. And this is what you can see on this Western blot. This is a MITF-specific siRNA that efficiently inhibits MITF expression. So of course, we worked with several siRNA to rule out non-specific effects, but today I show you data with only one siRNA. And so, for simplicity. And so what you can appreciate on these pictures is that in response to MITF knockdown, cells become flattened and enlarged. They display this typical blue staining that reflects the senescence activity. They also show these dots of heterochromatin protein 1 gamma, and this is the formation of senescence-associated heterochromatin foci. And here, you can see also that compared to control cells that proliferate over time till 6 days, here in these experiments, here the curve is flat, so MITF knockdown cells stop proliferating, and they are actually arrested in a G0, G1 group phase. We also assess the induction of apoptosis or cell death. So on this Western blot, you can appreciate that in contrast to TRAIL, which is a pro-apoptotic ligand of the TNF-alpha family that we used as a positive control here in these experiments. So TRAIL induced caspase activation, caspase or cysteine protease, which are activated during the process of apoptosis. So this is here the doublet. That is the active form of caspase-free, and here this is a cleavage of PARP, the poly-ADP ribose polymerase, which is one of the best known targets of caspase-free. And so you can see that there is caspase activation and cleavage of PARP in response to TRAIL. However, we cannot detect caspase activation or PARP cleavage in response to MITF suppression knockdown after 3 or 4 days. We also performed some FACS analysis, and we didn't detect any sub-G1 peaks, which reflect cell death after 3 or 4 days of MITF knockdown. So MITF knockdown cells display hallmark of cellular senescence, but no sign of apoptosis of cell death. So I also told you in the introduction that senescence is often associated with activation of the DNA damage response signaling pathway. So this is something we looked to. And so for those who are not introduced, very briefly, DNA damages induce the recruitment of sensor proteins such as the activated form of H2AX called gamma H2AX and 53BP1. that creates a sort of platforms to recruit effector proteins, such as ATM and CHEK2, that with P53 coordinate the cell cycle arrest and DNA repair, if it is possible, otherwise cells enter senescence or apoptosis. And so on these immunofluorescence experiments, you can see that a MITF knockdown triggers DNA damages, and the recruitment at site of damages of gamma H2AX, 53BP1, and the activated form of ATM and CHEK2 that are phosphorylated here and recruited at this site. What you can appreciate also on this Western blot is that a MITF knockdown is associated with a phosphorylation of P53 and serine 15, which induce P53 stabilization, and indeed we found an increase in P53 level in cells with a MITF knockdown. When we used inhibitor of the DNA damage response signaling pathway or a siRNA against P53, we dramatically inhibit the ability of melanocytic cells to become positive for the senescence-associated beta-galactosidase mediated by a MITF knockdown. So the DNA damage response signaling pathway is involved in the senescence program mediated by a MITF knockdown. What is interesting also to note is that this is something that we observe in our cell culture. So this is confocal images with the 3D reconstructions. So you can see that in control cells, chromosomes that are detected with phosphohistone H3 here in red, they are properly aligned along the equatorial plates. At the telophase, you can see here the two pool of sister chromatids that seems to be evenly divided between the two daughter cells. And here at the cytokinesis steps, you can see the mid-body that seems to be properly formed, which is detected with beta-tubulin in green. And in MITF knockdown cells, you can appreciate here some chromosomes that lags, that are not correctly aligned along the equatorial plates. You can see that we detected at the telophase here some chromosome lagging between the two daughter cells. And when cells finish to separate, this chromosome lagging are sometimes excluded into micronuclei. So we counted the micronuclei, and we indeed found an increased number of micronuclei in the cells with the knockdown of MITF compared to control cells. And this is a sign of genomic instability. And finally here at the cytokinesis step, you can see that the beta-tubulin is not localized at the most apical site of the mid-body, and so this will lead to defect aberration in cytokinesis. So to determine how MITF controls these processes, we combine an approach of RNA-seq experiments of MITF knockdown versus control cells with a genome-wide chip-seq of MITF. So this is example of those two experiments picture. And this, the combination of those two analyses allowed us to identify the full repertoire of all MITF direct and indirect target genes. And among the MITF direct target genes that are positively regulated by MITF, we found genes associated with cellular functions such as cell division, DNA repair, and DNA replication. So to sum up this part, this is a scheme that I would like to propose. So MITF knockdown trigger mitosis defects, the formation of nucleoplasmic bridges and micronuclei that leads to DNA damages and activation of the DNA damage response. I told you also that MITF positively controls some genes involved in DNA repair and replication, and so MITF knockdown should dampen this, the efficacy of the DNA damage response signaling pathway, probably amplifying some DNA damages that can also trigger some mitosis defects, and these damages converge towards P53 induction and senescence entry of melanoma cells and melanocytic cells. So this brings me to the second part of my talk on the MITF mutation that we identified. And to make a long story short, how we go to identify this mutation. So years ago, we identified MITF as a new substrate of caspases during apoptosis in melanocyte and melanoma cells. So what we did is site-directed mutagenesis to identify the caspase cleavage site in MITF. So we identify the caspase cleavage site. We so generated a non-cleavable form of MITF. We forced the expression of MITF in melanocytic cells, melanoma cells, actually. And what we observed is that the non-cleavable form of MITF render melanoma cells resistant to apoptotic stimuli. And so this prompted us to hypothesize that individuals who develop melanoma harbor a mutation in the caspase cleavage site, in MITF in melanocyte, leading to melanoma formation. And so we collaborate with a team in Paris to sequence MITF. So far we haven't identified a mutation at the caspase cleavage site, but we identify a mutation near this caspase cleavage site that changes the glutamate creating into lysine. So this is our study. So we found that the frequency of this variant is higher in patients who develop melanoma, whether single or multiple melanoma, compared to controls. So this variant has also been identified by other studies that are listed here with similar allele frequency. They also found that the variant, the MITF mutation, is associated with melanoma, whether single, multiple, but also familial cases of melanoma in this study, and with nodular melanoma in this study, which is a form of aggressive melanoma. So this variant confers a 2 to 5-fold risk to carrier of developing melanoma. And what is interesting also to note on this slide is that the MITF variant has been associated with a higher nevus count and fair skin in 2 studies, which are risk marker for melanoma. So of course, the first things we did is to determine the impact of this mutation on MITF. And so again, this is a schematic representation of MITF with all the post-translational modification reported. And 2 simulation sites, K182 and K316, were previously reported. So the glutamate creating is highly conserved among species from human to fish, and falls within the second simulation site here. And so the mutation changed this consensus simulation site, IKQE, into IKQK. That means that probably the mutation impact on the simulation of MITF. And this is indeed what we demonstrated. So here, this is a representative experiment performed in HEC293 cells in which we force the expression of MITF because MITF is not expressed in HEC293 cells. And we also force the expression of SUMO. So here, this is MITF that is detected as a doublet. This is a lower exposed blot of MITF. So when we force the expression of SUMO, we detected the simulation of MITF here and here. When we introduce a mutation of the first simulation site, we decrease the simulation of MITF. When we introduce the mutation, we identify in patient, we even more decrease the simulation of MITF. And we perform the double mutant, we abrogate the simulation of MITF. So more recently, we confirmed this data, this observation, in a more physiological settings, without forced expression of SUMO or MITF. So we perform proximity ligation assay in melanocytes that we isolated from patients with a wild-type form of MITF or from melanocytes with a mutant form of MITF. And so here, the red dots represents the association of MITF and SUMO. And this is what we detected in melanocytes with a wild-type form of MITF, but not in those with the MITF mutation. So having shown that the E318 lysine mutation impairs the simulation of MITF, the next question we had addressed is to determine the impact of this mutation on the cellular localization, stability, activity of MITF. So we found no change in the cellular localization or stability of this mutant form of MITF compared to the wild-type form, but what we observe is a change in its transcriptional activity. So this is a very simple experiment shown here that use a synthetic reporter vector that comprise 3 e-boxes, which are the MITF binding site, cloned upstream the luciferase genes. So what you can observe here is that when we co-express the wild-type form of MITF or the forms with a mutation in the first simulation site, we have similar transactivation activity of MITF, but when we introduce the mutation we identify in patient, we strongly stimulate the transcriptional activity of MITF. And this is in agreement with the well-known dogma of sumo-mediated transcriptional repression. So because this mutation stimulates the transcriptional activity of MITF, and because MITF binds to DNA, we perform a genome-wide analysis of the wild-type form of MITF and its mutant form. And so what this experiment shown here told us is that the wild-type form of MITF bind to about 9,000 loci, and the mutant form to about 22,000 loci, and the difference being a large number of weakly occupied sites by the mutant form of MITF. So this comparative data analysis also show that some sites are equally occupied by the wild-type or mutant form of MITF, such as in cluster E, for example, and some sites are preferentially occupied by the mutant form of MITF, such as in cluster D or C, for example. On the right here, you have some example of the preferential occupancy of the IF1 alpha, AP1, and ABCB5 loci by the mutant form of MITF compared to the wild-type. So I show you that E3 18 lysine mutation triggers MITF hypostimulation, change its genomic distribution, and change its transcriptional activity. So to assess the how MITF is related to the pathogenesis, how this mutant MITF is related to the pathogenesis mechanisms, we constructed a new Knokin mouse model harboring the MITF mutation from its own promoter. So these mice do not show growth abnormalities, no nevi or melanoma development within 24 months of age. The only phenotype we observed is a slight lightening of the color of the coat, the hair, and the tail. So this is here an example of Fontana-Masson silver staining of skin tail. So here in the mice with the wild-type MITF, you can appreciate the black melanin here detected by the staining, that are strongly reduced in the skin of MITF with the MITF mutation, and this is on the right, the quantitation of melanin. So this finding is actually in agreement with what was reported in some studies. I mean the association of the MITF mutation with fair skin. So next, what we did, we started to introduce the MITF mutation in 2 mouse models prone to develop melanoma. So the first model we used is a model that allows expression of oncogenic BRAF, specifically in melanocytes, thanks to the tyrosinase promoter after tamoxifen application. So this oncogenic BRAF mouse is nevogenic, it's known, and this is indeed what we observe. These mice develop nevi. But when we introduce a mutation of MITF, we significantly increase the number of nevi in this BRAF background, indicating that MITF is nevogenic. And again, this is in agreement with what was reported in some studies. I mean the association in human of the MITF mutation with a higher nevus count. Regarding melanoma development, you can see that these mice develop melanoma with a very long latency. It's about the blue line here, 400 days for the first mouse. And we found no change in the latency nor in the penetrance when we introduce a MITF mutation in this background. So what we did next is to introduce a MITF mutation in the second mouse model prone to develop melanoma, which allows the expression of oncogenic BRAF and loss of PTEN specifically in melanocytes after tamoxifen application. And here, this is a representative kinetics experiments showing that melanoma develop faster in presence of the MITF mutation. And for example, you can see here that after 26 days, in the mice with the wild-type form of MITF, they still show nevi that are very well separated, very well distinguishable. But here, those nevi already grew a lot and gather and form these tumors. So above, this is a Kaplan-Meier survival analysis that confirms this observation, showing that mice with a MITF mutation display shortened survival times compared to mice with the wild-type MRTF. So to determine at the molecular level how MRTF the mutant form of MRTF mediates its effect, we perform a transcriptomic analysis of 4 different tumors from those 2 groups. And what InGenV pathway analysis told us is that the MRTF mutation is associated with a decrease in expression of genes that are related to senescence, such as CDKN2A, CDKN2B, and CDKN1A. And this indicated that the MRTF mutation decrease the process, the ability of cells to enter senescence. So here, this is the hypothesis we tested. We used human normal melanocyte isolated from patients with a wild-type form of MRTF and with a mutant form of MRTF, in which we force the expression of oncogenic form of BRAF. So you have here the expression of the oncogenic form of BRAF, which translated into a similar activation of ERK downstream. And what you can appreciate here is that in a melanocyte with wild-type MRTF, the oncogenic form of BRAF is able to induce this typical blue staining that reflects senescence entry of melanocytic cells. But melanocyte with a mutant form of MRTF display a delay in the ability to enter the senescence process. So the MRTF mutation seems to impair oncogene-induced senescence in normal human melanocyte. So to sum up, this is the schema I would like to propose. So melanocyte can give rise to nevus or senile lentigo, and this is related to senescence, premature senescence, replicative senescence, or aging. And our work showing that MRTF inhibition can trigger senescence could be of interest in this process. And more globally, we identify the full repertoire of MRTF target genes, among which we find some genes that regulate pigmentation and the senescence process. And our finding can also find interest in the progression of those lesions to melanoma. So I told you that nevi are considered the very best example of senescence in vivo. They can remain static for decades and never progress towards malignancy unless other alteration occurs. That leads to senescence bypass. And with this senescence bypass, cells restart their proliferation with less checkpoint control, so genomic instabilities occurs. And with reacquisition of pseudo-epithelial mesenchymal transition, cells start to disseminate, form mets, and usually these metastases are drug resistant. And so our finding could find interest in these 2 processes to improve anti-melanoma therapies. So to finish, I would like to thank our sponsor. And I would like also to thank all the people in the team who, and some who participate to that work. And I would like, also, of course, to thank Robert. And this is here our collaborator in France or abroad. Thank you very much for your attention.
Thank you very much, Corinne. Is there any question?
The first mice with the mutation, you said that this mice has some code color dilution. And if I understood well, these mutants have some more transcriptional activity, more binding to target. So I'm not sure to understand the relation between the 2. So if you can just comment on that.
Yeah, okay. So regarding the first question, we have lots of genes involved in kinetochore in the list we identified by our RNA-seq and chip-seq analysis of MRTF. So for that, it's we don't have only one. So we have several genes involved on those process of replication and the DNA repair and kinetochore formation, etc. So yes, this fits with the phenotype we observed. Regarding the regulation of pigmentation by the MRTF mutation, it's true that we observe an increase in transcriptional activity, but it's a transcriptional activity, it's a global transcriptional activity on on e-boxes that are not in their genomic environment. And so we know that MITF is able to bind to genes that are not related to pigmentation, but that can affect pigmentation. And we know that MITF can bind to DNA and exert some repressional transcriptional activity. So I guess there is a mix that we don't know for the moment between those activities that leads to a decrease in pigmentation. But
yes,
actually, Cedric, the regulation of pigmentation is not huge. It's a slight lightening. So we don't know because we didn't succeed at the lab to stain, to know if it's a change in the activity of melanocyte or in the number of melanocyte in the skin. So I have no answer for the moment to that.
Corinne? Perhaps there are something I do not understand exactly, but you have shown that the mutant MITF delay senescence induced by RAS or RAF.
BRAF, yes.
In vitro, but in vivo, you have shown that there are more NEVI. Is it correct?
Yes.
So I don't really understand. In vitro, it delays the occurrence of senescence, and in vivo, it induces more NEVI.
Yeah. Because it's true, you're completely right. I show two things. I show that indeed there is an increase in the number of NEVI, and what we show is that the MITF mutation, the melanocyte with the MITF mutation, have a doubling capacity that is higher than normal melanocyte. Okay, we have that. So they proliferate a little bit faster, and I think that's why we saw an increase. But after that, they are able to stop. There are other checkpoint control that allow them to stop. So indeed, in the oncogenic BRAF background, we show that. In the BRAF PTEN background, we show that NEVI actually they progress towards malignancy, towards formation of melanoma, and they don't stop because you lose also another checkpoint here, and so they are able to move towards melanoma. And in the culture conditions that we used, we also have this increased activity in the PI3 kinase pathway in the medium that we used with the MITF mutant, and we think that that's why we observe this ability to delay the capacity to enter senescence.
Very nice presentation, many thanks. I was wondering about the influence of the simulation versus a potential ubiquitination at that site, and if that would also impact on the stability of the protein on the one hand, and that then would lead to a second question about have you ever tried to overexpress MITF in melanocytes, and would that then be prolonging the replicative lifespan and be protective? Could that be a target for increased activity as an anti-tumor mechanism?
Okay, so for the first question, you're right. So the mutation changed glutamate into lysine, so it's not nothing, and we lose the simulation for sure, but perhaps it's not directly the simulation impact that affects all the processes we looked at, but perhaps it's a change to ubiquitination. You're completely right, and we haven't studied these mechanisms, so we don't know, but we know that simulation is affected by the mutation. And for the second question, yes, we also have a delay in the senescence when we force the expression of MITF, but you can have also regulation of simulation independently of the mutation. So. But the senescence bypass is higher with the mutant form of MITF.
Okay, I have just a short question for you before you leave. I'm wondering if you, in the list of your protein interacting with MITF, you don't have lamine A or lamine C, because in diseases with mutation of the gene LMNA encoding these proteins, you can have trouble of pigmentation. You have hypo and hyperpigmentation. So, and you have also alteration of the nuclei of the cells. So I was wondering if you have found this protein.
I don't know I don't know precisely the list of all MITF target genes, but I can check, and there are many nuclear proteins. So perhaps there are lamine C and A.
And A, yes.
I can check for you if you want.
Okay, thank you very much. So now we welcome Professor Corinne Abadie. She's coming from the opposite of Nice, from Lille in the north of France. She's working in Institut Pasteur de Lille, and she's a specialist of senescence in different type of cells. And she will speak today on senescence of skin keratinocytes and fibroblasts, and she like initiation of cancer.
Thank you very much, Frédéric, for this invitation in this so beautiful place. So I will talk indeed about the mechanism of senescence of skin keratinocyte and fibroblast. So the introduction of senescence was done by Corinne very well, but perhaps I will introduce again the mechanism. So senescence was defined historically as a state reached by cell after an exponential growth phase in vitro culture. And at this growth arrest phase, cells undergo several alterations. First of all, they have a very different morphology from growing cells. They are bigger and spread on the dish. They look very damaged, but actually they are perfectly alive, and even they are apoptosis resistant. So they are cell cycle arrested, mainly in G1, and they display several other modifications in metabolism, for example. They change the composition of their secretome, which becomes pro-inflammatory. They also display several epigenetic alterations, and they suffer from oxidative stress and endoplasmic reticulum stress. And in consequence of these stresses, there is an increase in autophagic activity, and actually, I think it is used as the very well-known marker of senescence, the SB marker. So, as Corinne told you, senescence can be induced as a result of shortening and dysfunctioning of telomeres. And now this type of senescence is referred as replicative senescence. But senescence can also be induced by several stresses: oxidative stress, oncogenic stress, and also stress related to some therapies, including anti-cancer therapy, such as some chemotherapy and radiotherapy. The common point to all these subtype of senescence is that there is a formation of DNA damage, and this DNA damage induces the DNA damage response pathway, which leads to the stabilization and activation of the tumor suppressor gene P53 and one of its target, P21, which is a cell cycle inhibitor, and so leads to the typical cell cycle arrest of a senescent cell. Actually, it is known also that the P16RB pathway is often involved in the cell cycle arrest of senescent cells, as well as the P38 MAP kinase pathway. So, regarding senescence of dermal fibroblast, what is clear when it occurs as a replicative senescence is that this senescence is irreversible. I think it is quite clear on this culture. We did this culture for about 2 years, and during this time, the senescence growth plateau was very stable without any obvious proliferation nor any obvious cell death. But in my team, we are interested in the relationship between senescence and tumor initiation. So with this type of results, it is assumed that senescence behaves as a tumor suppressor mechanism. But perhaps it is not so relevant to work with fibroblast as a model of senescence when you are interested in cancer initiation. So in my team, we decided to study the mechanism of senescence of epithelial cells, and we choose this model of skin keratinocyte. So with skin keratinocytes in culture, cells also undergo senescence, but one can say that this senescence is premature compared to fibroblast in the previous slides. You can see that with fibroblast, senescence occurs after 5 to 6 months of culture in vitro and after about 50 population doublings. And with keratinocytes, senescence occurs much more prematurely after just 2 to 3 weeks of culture and about 20 population doublings. But the cells are indeed senescent with all the markers of senescence. And another big difference with fibroblast is that this senescence is not irreversible. We found that there are 2 possible outcomes from senescence for keratinocytes. Most of cells die, but they do not die by apoptosis. They are apoptosis resistant. They die following a very huge increase in the autophagic activity. Just an image to show this autophagic activity. This is an electron micrograph of a dying senescent keratinocyte. And you can see all this accumulation of autophagic vesicle around the nucleus, which is itself very altered. So most of the cells die by autophagic cell death, and only a small fraction of cells, about 1 cell on 10,000, is able to re-enter cell cycle to generate clones of new daughter cells that reproliferate in the culture dish. So we spend a lot of time to characterize these post-senescent cells, and we have shown that they have undergone an epithelium to mesenchyme transition. For example, here with this increase in twist, which is a transcription factor which play a major role in epithelium to mesenchyme transition. For example, also this decrease in the expression of MET, which is a receptor specific of epithelial cell. We also performed transcriptomic analysis, which show that about 40% of the genes of the transcriptomic signature of these post-senescent cells are common with genes activated in cancer. And also, importantly, we have done a tumorigenesis assay, and we have shown that these post-senescent cells generate some skin lesions which were characterized as a small hyperplasia or small carcinoma. And also, very importantly, we performed HPRT assay to look whether these cells were mutated. When cells are not mutated as normally growing keratinocytes, they are sensitive to the toxicity of 6-thioguanine, as you can see here. But the post-senescent emergent cells are resistant to the toxicity of 6-thioguanine. So we conclude that these cells are tumoral, or at least pre-tumoral. So with keratinocyte, one cannot say that senescence is tumor suppressive, but perhaps rather a step that prepare to tumorigenesis. And so we used this model of cell culture to study the very first event at the origin of tumor initiation. So to make this study, we performed a comparative analysis of the mechanism of senescence of normal human epidermal keratinocytes and normal human dermal fibroblasts. And we worked with cells from the same donor to avoid any differences to polymorphism. And so the first things we looked at was the telomere shortening by a fish analysis. So as largely published by others, we confirmed that at senescence in fibroblasts, there is a shortening in telomeres, which is quantified here. But at senescence in keratinocytes, the length of the telomeres remain constant, and there is only a very non-significant decrease. So senescence of keratinocytes is not replicative senescence due to telomere shortening. So we looked also at the activation of the DDOR pathway. So we looked at the recruitment of most of the protein of this pathway at foci of DNA damage. So as you can see, in fibroblasts at senescence, the pathway is activated, but it is not activated at all at senescence in keratinocyte. And in consequence, in fibroblasts at senescence, we confirmed that there is a stabilization and an activation of p53 and an upregulation of p21. But at least at the beginning of the senescence plateau in fibroblasts, the p16RB is not involved. Actually, it is involved in deeper senescence, but not at the beginning of the plateau. And in comparison, in keratinocyte at senescence, there is no activation of p53 and p21, but there is an upregulation of p16 and an hypophosphorylation of RB. So the cell cycle arrest pathway involved at senescence in the two types of cells is different. So the question was, but why senescent keratinocytes stop proliferating? What are the damage, the DNA damage that could be at the origin of the cell cycle arrest? And to answer to this question, we used a COMET assay. COMET assays are electrophoresis performed on single cells. If in the nuclei in the cells there are some broken DNA, it will migrate to form this comet tail that can be measured. So if you performed a COMET assay at pH 8, the broken DNA that migrate originate from double-strand breaks, and if you perform the assay at pH 12, the two strands of DNA are separated, and so the DNA fragments that migrate originate from both double-strand breaks and single-strand breaks. And so at senescence in fibroblasts, we detected the presence of DNA double-strand breaks, as it was already described by others, but no double-strand breaks at senescence in keratinocytes. And in contrast, we found some single-strand breaks present at senescence in keratinocytes. So there are some specific DNA damage, single-strand breaks, occurring at senescence in keratinocytes. So a few words on the signalization pathway induced by the presence of DNA single-strand breaks. So the breaks are first recognized by the PARP1 enzyme, the poly-ADP ribose polymerase 1, which will polymerase a network of poly-ADP riboses. This network recruits the XRCC1 protein, which in turn recruits the repair enzymes themselves. So to confirm the COMET assay, we looked at the formation of XRCC1 foci at senescence using 3 different antibodies recognizing 3 different epitopes. And so as you can see, there are an accumulation of a lot of XRCC1 foci at senescence in keratinocytes. This is quantified here. And there are also accumulation of XRCC1 foci at senescence in fibroblasts, but the accumulation is less important. And you can also see that the foci are smaller than in keratinocytes. This is especially evident with this antibody. So from these results, we suspected that there could be a problem with the repair of DNA single-strand breaks in senescent keratinocytes. And so to look at that, we first evaluate the expression of PARP1, and we saw by RT-PCR that PARP1 expression is decreased at senescence in keratinocytes, whether it is maintained at senescence in fibroblasts. This is confirmed here by Western blot, the decrease in keratinocytes at senescence and the maintenance of the expression in senescent fibroblasts. And so in consequence, there was also a decrease in the PARP1 activity, the formation of poly-ADP ribosis in senescent keratinocytes compared to senescent fibroblasts. So if PARP1 activity is decreased, the breaks could be not repaired, and it is the case indeed. We show that using an incorporation of BRDU, because when the repair occurs, there are some neosynthesis of DNA. So you can see that in fibroblasts, there is an incorporation of BRDU at the localization of the DNA damage, but there is no formation, no DNA synthesis in senescent keratinocytes. And also, we confirm that by looking at the recruitment of the ligase. So the ligase is recruited in the XRCC1 foci in fibroblasts, but not in senescent keratinocytes. So the SSBR pathway is defective in senescent keratinocytes because of a decrease in PARP1 expression and activity. So the next step was to determine whether this default in the SSBR pathway was responsible for the upregulation of P16 and the cell cycle arrest. So to answer to this point, we re-expressed PARP1 using an adenoviral vector in already senescent keratinocytes. So you have here the level of re-expression, and this re-expression of PARP1 induced a decrease in the expression of P16 and restored a normal phosphorylation of RB. Sorry for the text, there is a problem with the slide. We also looked whether the abnormal, very big XRCC1 foci were at the origin of the upregulation of P16. And so to look at that, we performed an invalidation of XRCC1 using scRNA. So you have here the level of invalidation. And when XRCC1 was invalidated, there was a decrease in P16 and a restoration of RB. In these two experiments, there is no change in the expression of P53 nor P21. And finally, we asked whether the P38 MAP kinase could be involved in this pathway. And so upon the invalidation of XRCC1, indeed there is a decrease in the phosphorylation of P38 MAP kinase in conjunction with the decrease in the expression of P16. So we have at senescence in keratinocyte the activation of a pathway that is induced by the abnormal accumulation of XRCC1 at the foci of single-strand breaks, which will lead to an activation of the P38 MAP kinase and then to the upregulation of P16. So this is the pathway involved in the cell cycle arrest of senescent keratinocytes, but is this accumulation of unrepaired SSB involved also in the post-senescent neoplastic emergence? It was our hypothesis because the accumulation of unrepaired DNA damage could be mutagenic. So to assess that, we induced a premature downregulation of PARP1 in proliferating keratinocytes using 3 different siRNA. And as you can see, this downregulation of PARP1 induced a premature senescence plateau, followed by a post-senescent emergence. So these 2 events are correlated with the accumulation of XRCC1 foci, the accumulation of unrepaired single-strand breaks, and no change in damage able to induce the DDR pathway. These post-senescent emergence obtained in this situation are similar to that obtained in control cell. For example, you have again a downregulation of the expression of MET, an upregulation of this receptor, which is specific of the post-senescent emergent cells, and these cells are also mutated as the normal emergent cells. So one question also was what is the inducer of the single-strand breaks? PARP1 induce an accumulation of this break, but do not induce the formation of the breaks. So, of course, our hypothesis was that the break were the consequence of the increase in the oxidative stress associated with senescence. So, to look at that, we treated cells with antioxidants. And as you can see, when we do that on pre-senescent cells, we reduce completely the accumulation of XRCC1 foci and the accumulation of unrepaired SSB. On the contrary, if we applied an oxidative stress to proliferating cells, we induce an accumulation of XRCC1 foci. And the quantity, the concentration of H2O2 we used was not enough to induce double-strand breaks able to activate the DDR pathway. So it was quite expected, but what was less expected was that the oxidative stress is also the inducer of the downregulation of PARP1. So you can see here that if we treat senescent keratinocyte with N-acetylcysteine, which is an antioxidant, we restore the expression of PARP1. And on contrary, if we treat growing keratinocyte with H2O2, we induce a decrease in the expression of PARP1. And so in consequence, if you treat keratinocyte with antioxidant, you delay the occurrence of the senescence plateau compared to control cells, and you suppress completely the post-senescence emergence. And in contrary, if you treat proliferating cells with H2O2, you induce a premature senescence plateau followed by a post-senescence emergence. So all these results also, of course, were in vitro results. So we wanted to establish whether this mechanism of senescence and post-senescence emergence were true also in vivo. So for that, we used human skin samples from old people. It was about people from 70 to 80 years old, compared to young people about 30 years old. So we performed the analysis on the epidermis. The word is lacking. On the epidermis or in the dermis. So on the epidermis, we search for an accumulation of XRCC1 foci. And as you can see, there are a lot of cells in the epidermis that are positive for XRCC1 foci. This is quantified here. About 40% of the cells accumulate this foci. In contrast, there is absolutely no accumulation of DDR foci. In conjunction of this accumulation of XRCC1 foci, there is a sharp decrease in the expression of PARP1 in about, you go from about 60% of cells that express PARP1 to only less than 10%. And in conjunction with this decrease in PARP1 expression, there is an increase in the oxidative stress shown here by the expression of the SOD2 enzyme. And in contrast, in the dermis, sorry, it lacks a slide. In the dermis, in contrast, there is no accumulation of XRCC1. There is no XRCC1 foci in these cells. But in contrast, there is an accumulation of DDR foci. I think you can see 1 or 2 spots in the fibroblast in the dermis, in about 20% of the fibroblast of the dermis. There is no change in PARP1 expression, and there is also no change in oxidative stress measured by this expression. So actually, in vivo, the cells accumulate with aging the same type of damage than in vitro senescence. So to conclude, we propose that there exist 2 types of senescence in human skin. One concerns the fibroblasts, who undergo probably replicative senescence, so classical replicative senescence with telomere shortening, accumulation of double-strand breaks, which would lead to an upregulation of P53 and P20N, and to a very, very stable tumor suppressive senescence. But in keratinocytes, and actually in other epithelial cell types, such as mammary epithelial cell types, senescence is due to oxidative stress. There is no telomere shortening, no generation of double-strand breaks, but there is a decrease in PARP1 expression, which leads to the accumulation of unrepaired SSB, which will lead to the activation of the P38 MAP kinase and to the P16 cell cycle inhibitor. But this senescence is not so stable. Most of the cells will die following the huge oxidative stress by autophagic cell death, and some rare cells are able to re-enter cell cycle and generate a progeny of mutated cells that could be at the origin of a skin cancer. Okay, I would like to thank all the team who performed the work, especially Joe Nassour, who does most of the experiment I've shown today. Thank you very much.
Thank you, Corinne. Is there any question? Yes. Corinne.
Thank you very much for this very nice presentation. I would like to start with a question to one of your last slides, meaning the XRCC1 foci in the epidermis.
Yeah.
So it appeared that you saw foci also in the suprabasal differentiating cells. Do you have any concepts of what would be the functional reason for cells that undergo terminal differentiation anyway to have, say, a stress-inducible DNA
actually we have looked whether the accumulation was in the basal layer or in the suprabasal layer, but there was no clear difference. So
because, I mean, for say the basal cell layer, it would seem very important, but is there any effect, say, of XRCC1 knockdown on the differentiation properties of keratinocytes in culture? So if you expose them to
Yes, we haven't looked at that. Just what I can say is that in vitro differentiation induced by calcium addition into the culture medium, senescent cells, senescent keratinocytes are able to differentiate in this condition. So with their XRCC1 foci, they are able to differentiate. But it was only in vitro. In vivo, I don't know.
Corinne?
Corinne, do you have any details about the regulation of PARP level by oxidative stress?
No. We have to do that. And it's quite surprising. We are very surprised because it's not intuitive to understand that upon a stress that induces single-strand breaks that have to be repaired by PARP1, the same stress also induces the decrease in the repair enzyme. So it's difficult to understand. It's a fact. And the pathway, no, we don't know. Actually, it's the pathway. We have other experiments. I didn't show them, but the pathway is really activated by the presence of this abnormal, very big XRCC1 foci. And we don't know how this big foci could lead to the phosphorylation of the P38 MAP kinase. This is the first step we have to understand. And then we have to understand at the promoter of P16 what is the relationship between P38 MAP kinase and the promoter of P16.
If I may, I have another question. Yeah, I was wondering also if you know the mechanism by which those few cells escape the autophagic death mediated by autophagic stress. It's because those cells have some repair mechanisms that are more developed. Or...
Okay, so of course it is the big question. So you are right. The cells that re-enter cell cycle to generate the progeny of mutated cells have escaped autophagic cell death. And for this specific point, we have shown that it depends on the level of the autophagic activity. So we have formally shown that the cells that have a level of autophagic activity that is just slightly lower than that will die are those that have the higher probability to re-enter cell cycle. And this level of autophagy is directly linked to the level of oxidative stress. So the cells that have the higher level of oxidative stress die by autophagic cell death, and those that have a level just slightly lower, but which is much higher than in a proliferating cell, 20-fold higher approximately, are those cells that re-enter cell cycle.
Lionel.
So, thank you for your call. Just definitely, it seems to be difficult to have a control of this kind of system through induction of repression of P16 or P21 in other kind. But when you mention in the conclusion that P60 is induced by oxidative stress, do you know which kind of specific part of the oxidative stress is involved or not?
Yeah. So previously we have shown that the senescence growth plateau of keratinocytes is associated with an activation of NF-κB, transcription factor, and SOD2 is a targeting of NF-κB. So there is also an upregulation of SOD2. So SOD2 is described as an antioxidant enzyme, which is involved in the dismutation of O2 point minus into H2O2. But actually, what we have shown is that there is a a disequilibrium in the antioxidant pathway induced that way, because if you have an upregulation of SOB2 without any upregulation, for example, of catalase or glutathione peroxidase to degrade H2O2, you have an accumulation of H2O2. So you have a decrease in O2 point minus, but an accumulation in H2O2. And this is one of the pathways involved in the increase in oxidative stress in keratinocyte senescence.
And did you try to test some compounds to involve or to modify your process?
Yes, so with the, for example, as an antioxidant, we use catalase, which is specific for the degradation of H2O2, and catalase delay the occurrence of the senescence plateau and delay or decrease actually the emergence frequency.
Professor Grillari.
Wonderful presentation again, thank you.
Thank you.
If I remember correctly, Jerry Shea and Woody Wright proposed a model for epithelial cell senescence, I don't know, 20 years ago, claiming there is a mortality stage 1, that is telomerase independent, and then after this escape that you observe, they eventually enter something like permanent growth arrest and don't form tumors after that. So my question is, did you follow up on the cultures after they escaped this first plateau that you described?
Yeah. So actually, these cells are not immortalized. They undergo a second growth plateau, but this is still not telomere dependent. Telomeres are still quite long at this plateau. There is some small shortening, but not enough to induce replicative senescence. So we didn't work a lot on this second growth plateau, but what we did was to measure the level of oxidative stress at this plateau, and actually it is higher than here. So this second plateau is also oxidative stress dependent. There is also an upregulation of P16 at this second plateau. And the morphology of the cells is very similar to the morphology of senescent cells, but it is some way a bit different, but I can't describe it very clearly. It's very similar. And we can have a second emergence from this second plateau. But the frequency of this second emergence is lower. It is perhaps about 1 cell on 100,000. So it's difficult to have an in vitro culture. You need a lot of boxes of culture to see that. But when there is this second emergence, the second emerging cells are much less transformed than this one. So actually it is a mechanism step by step. Perhaps we can imagine that the second emergent cells could enter a third plateau, but we have never observed that because we never wanted to do that. But this is the idea. Perhaps you have step by step an increase in the tumorigenicity of the cells.
Thank you. Thank you very much. So now we will welcome the last speaker of this session, Apoptosis versus Senescence. It's Professor Johannes Grillari, who is coming from the Institute of Biotechnology of Vienna. So, grüß Gott, Herr Professor Johannes Grillari. And he's a specialist in senescence, aging. It's hard to describe him because it's at the same time molecular biologist and biochemist and geneticist. And today the title of his talk is Skin Cell Senescence, a driver of skin aging and how to drive back. So we are happy to know the answer.
Merci bien, grüß Gott, bonjour. It's a great pleasure to be here for the first time in this wonderful location. Actually, I think I should sing my presentation at this opera, but I guess then you would run for coffee right now and not after my talk. As a disclosure, I'm on the one hand, I'm still an academic working at university, but I'm also co-founder of two companies, Eversight and Tamerna. And my lab is in part co-funded by Chanel Research and Technology. And that's part of what I'm trying to tell you today. So my lab for the last, and I don't dare to say that, for the last probably 20 years has been focused on understanding why we have differences in the speed of the aging process. So what drives biological aging? And I think this is a wonderful example of the difference in the speed of the aging process. These samples are normalized for social background. Both are musicians. You have Tina Turner at the image taken at the age of 68 versus Keith Richards at the age of 65, and you can clearly see, especially from a dermatological perspective, that there are differences in the speed here. And probably, and that's one of the ideas we had in the lab, maybe senescence is one driver of premature aging of this guy. Might also be the substances that run in the veins here. We don't really know. So you already heard the definition of cellular senescence. Just to recapitulate, it's been an in vitro phenomenon for a long time. You explant the cells from the human body, get them into culture, and eventually after reaching a certain number of population doublings, this number is termed the Hayflick limit after Lenny Hayflick, who first described this phenomenon. The cells enter this growth arrest, but they can do that also earlier if they are hit by a stressor, as we heard before, by oncogene, by hyper-oncogenic signaling, or by oxidative stress, for example. And I think also by there's something now termed something like a mechano-induced senescence by just having not right adherence factors on the cell culture plates. Very in vitro, but in vivo, how does it look like? And by now, we have evidence from several labs, from several different studies, that senescent cells accumulate in the human body at a lot of different sites, different tissues, different organs, and especially at sites where we see age-associated disease-dependent lesions in the brain, in the lung, in the kidney, in the bone, and we also have estimates from a couple of different studies of about 20 to 50% of senescent cells that we find in the skin of elderly. So can we now imagine that such senescent cells really are not only involved in suppressing tumors or promoting tumors per se on a cell autonomous level, but do they really have something to do then with aging? Because we expect that they would, if they are a driver of aging, then that they would somehow impact on the organismal aging process. And there have been a couple of papers out now from specifically from the Mayo Clinic, Jim Kirkland's lab and Van Dersen's lab, as well as from Buck Institute, from Julie Campisi's lab, that show that if senescent cells can be eliminated in mouse models, and these are, by now we have transgenic mouse models, and we have also mouse models where senescent cells are eliminated by so-called senolytics, drugs that specifically eliminate and drive senescent cells into apoptosis, that then we have postponing of almost all of the major age-associated diseases, including osteoporosis, lung fibrosis, osteoarthritis, atherosclerosis. We seem to rejuvenate hematopoietic stem cells. We have a reduction in the side effects of anti-tumor therapy. We saw that we have something called chemotherapy-induced senescence as well. And there's a paper, one of the early ones, that also shows beneficial effects of senescent cells. So in contrast to the other studies by Judy Campisi's lab, we see that if we would kill senescent cells during wound healing, then we delay the closure and the healing of the wound. On the other hand, we also see that if we have persistent senescence in the wounds, then we also impair differentiation of keratinocytes, and wound closure is also not working that well. So we have need a balance. So how does that look like if we kill senescent cells in a mouse model? This is from Peter de Keyser's lab. He is now in Utrecht. This is the treated mouse at the same age as here the untreated mouse. The elimination, the senolytic drug used here is a peptide that inhibits p53 interaction with FOXO4, which is a specific interaction that happens in senescent cells. And I think this closely resembles to Tina Turner versus Keith Richards here. So why are senescent cells then bad in the sense of driving the aging process? Whatever the trigger for cells to go into senescence might be, on the one hand, and we heard in the previous talks about it, they stay within the tissue, they alter their functionality, they alter the differentiation status, they might be endogenously forming tumors later, might be. They also get killed by the immune system or by apoptosis in some instances. So they use up the replicative potential of the surrounding tissue, and this is what my lab at the moment considers as one of the important drivers in the aging process, is that they accumulate something like a pro-inflammatory phenotype, the so-called senescence-associated secretory phenotype, or SASP, which is considered one of these antagonistic pleiotropies, meaning that the senescence per se might be a tumor suppressive mechanism, while on the other hand, this pro-inflammatory phenotype, the metallomatrix proteinases that are secreted as well, which remodel the stroma, that this might be actually even pro-tumorigenic, and therefore might also drive this tumorigenesis in this opposite way as we would conceive senescence per se. So my lab started out to look for factors that are differentially expressed in early passage versus senescent cells, and we wanted to see if such factors then really can impact on the on the organismal lifespan. And so after quite some time, we came up with at least two factors that we identified in such screens that actually do have an impact on the organism lifespan. Here's one, it's a DNA damage repair, splicing and ubiquitination factor called SNEF by our lab, HBRP19 or PSO4 by other labs. If we overexpress that dose dependently in flies, then we indeed increase the lifespan of these flies. The other enzyme that we identified is an ribosomal RNA methyltransferase that if we knock it down in fly, worm, or even in yeast, can expand the lifespan of these organisms. In that case, so we hit on a novel conserved pathway of organismal longevity by looking at cellular senescence. So what did we do in terms of skin aging? So we know, and that might be questioned, but there are papers claiming that there are up to 50% of senescent cells in human geriatric skin. So we tried to model this situation by generating 3D full thickness skin equivalents where we mix senescent fibroblasts into the collagen matrix and then seed the keratinocytes on top and have them differentiate. And you will see more about this model in a talk after the break by a fantastic, actually she's still a PhD student in the last days, very close to being a postdoc in my lab, Regina Wein-Müllner. And so she will tell you a bit more about this model. But let me just say at this point that when we add senescent fibroblasts into this model, then we definitely see a reduction in the epidermal thickness that comes along. So can we now somehow, because obviously there must be a crosstalk between the fibroblasts that are inside of these of the collagen matrix to the keratinocytes and an impact on the differentiation process of these keratinocytes. So can we somehow alleviate this not so good effect of senescent cells? And in our collaboration with Chanel Research and Technology, we screened a couple of plant extracts for their effects on senescent fibroblasts in that case. And we came up with one, Solidago alpestris, Solidage or goldenrod, is this yellow flowers that we have, an alcoholic extract of that impacts on senescence. So how did we find that? We screened a couple of different of these different plant extracts on our fibroblast senescence model, and this extract 1201 from Solidago decreased already in early passage and midterm passage cultures the amount of senescence-associated beta-gal positive cells. So we zoomed into that one and stayed with that extract, and especially looked at the effect on the SASP, on the secretory phenotype of after being treated with 1201 extract. And what we indeed see is that the pro-proliferative effect, this potentially pro-tumorigenic effect of senescent cell secretomes is dampened by 1201. And we tested it by taking conditioned media before and after exposing them to 1201 onto pre-neoplastic cell line HACAT cells, and we see that we have lower proliferation of these cells. We also see that the chemoattraction, which is also known to be a functionality of this SASP, is dampened by exposing the cells and treating the cells with this plant extract. So what happens now if we put the plant extract onto senescent fibroblasts before we then include them into our 3D skin equivalent? And what happens is that we can beautifully restore the differentiation process of the keratinocytes. So meaning that indeed whatever is happening in terms of dampening the secretome of senescent fibroblasts is also active for a couple of days after being treated, and then transferred to the keratinocytes in the 3D skin equivalent. And obviously, because senolytic activities of a very high interest in the field at the moment, we tested if 1201 would then also have a senolytic activity by specifically killing senescent cells in vitro, and we find yes, it does. It does so very slowly and very lowly, but significantly after exposing senescent cells for 39 days to this Solidago extract. then we have about 30% of senolytic activity that we can observe. So it's not a huge and big senolytic that might have a huge clinical potential, but still there is something in there that where we were very happy about it. So now, what is this SASP? What do we find in there? It's pretty well known that we have a lot of pro-inflammatory cytokines like IL-6, IL-8. We have matrix remodeling factors like MMP1, M3. We also see some eicosanoids, prostaglandins in there. But we don't really know about extracellular vesicles and microRNAs. So one question that we had in my lab is, do we find extracellular vesicles and their microRNA cargo as one component of the SASP? So what are extracellular vesicles? A very hot topic at the moment. The Society of Extracellular Vesicles now tried to counteract the Babylonian language problem of the naming of these vesicles coming down to 3 names only, depending on the biogenesis. If the vesicles come from the endosomal compartment via multivesicular bodies, then they are called exosomes. If they bud from the plasma membrane, you call them ectosomes or microvesicles. And if they are blabbing from apoptotic cell membranes, then you call them apoptotic bodies, obviously, so that name did not change. So that's what we call them in the field now. And any known uptake mechanism that has been described before has also been shown in the context of taking up extracellular vesicles. Might it be phagocytosis, fusion to the membrane, endocytosis? All has been published in the context with different vesicles from different cell types being taken up by other cell types. So, since the biomarkers for the extracellular vesicles are not very clear yet, and we have proteins from the endosomal compartment that have also been found in the ectosomes, and we still rely on size as a marker for the EV species, which is not a good marker, but that's how we fractionate and purify the vesicles. If they are below 100 nanometer in diameter, we call them exosomes. If they are 100 nanometers to 1 micrometer, we call them microvesicles. For apoptotic bodies, they are usually larger, and they get annexin 5 positive by exposing phosphatidylserine to the outside of the membrane. So we consider these extracellular vesicles as messages in a bottle. So you have the double lipid membrane layer around it, and then you have some kind of a letter inside of the extracellular vesicles. And the question is, what is written on this letter? And by now we know that the letter tells the recipient cells a lot of different things in terms of growth, proliferation, angiogenesis, inflammation, immune response, epigenetic reprogramming, invasivity. It prepares the premetastatic niches at distant sites from the primary tumor. So there's a lot to say on these letters, and the question is, what is the language the letter is written in? And by now we know that this language comes in the flavor of the amino acids, meaning proteins. On the other hand, on the letters of the genetic code, meaning nucleic acids in form of RNA and DNA, but also in terms of lipids, which are transported by extracellular vesicles. And we focused on microRNAs in that regard. So first we tried to see if extracellular vesicles would change their shape and distribution between senescent and early passage control fibroblasts. They do not. The shape is pretty similar to what you would expect from these extracellular vesicles after you purify them and look at them in transmission electron microscopy. Markers like TSG101, which is an exosomal marker, is present. And we see that indeed senescent cells, and in this case we used oxidative stress-induced senescence on fibroblasts, is the vesicle secretion goes up by between 3 and 6-fold depending on the donor and the repetition of the experiment. With that, obviously, we have an increase of all the cargo molecules that are inside of the vesicles in terms of microRNAs. So almost all microRNAs are upregulated. If you look at the global level of the total amount of vesicles you have per cell that are secreted, but then we also find some specific changes in the composition of the single vesicles, which I don't tell you now. So since everything is up, what are the top 20 secreted microRNAs? We made a list and we performed pathway analysis, and what comes out is that most of the direct targets of these microRNAs seem to hit on the apoptosis behavior of other cells. So the logical experiment was then to take the extracellular vesicles from senescent cells versus control cells and see if we would, in that instance, decrease the apoptotic behavior of the recipient cells. So the experiment we performed was then really showing that we have a reduction in the apoptotic behavior of fibroblasts that were acutely stressed by hydrogen peroxide, which drives them into apoptosis. So higher amounts, not senescence-inducing amounts of hydrogen peroxide, but really apoptosis-inducing amounts. And with the vesicle from senescent cells, we indeed decrease the rate of apoptosis of the bystander cells. So this might be something bad, again, in terms of tumor development, where senescent cells might decrease the ability of damaged cells in the surrounding to undergo apoptosis, and therefore might allow some mutated cells to survive and eventually form a tumor here. So are now the microRNAs really transferred from the keratinocytes within a 3D experiment, a skin equivalent experiment? Are they transferred to the keratinocyte, to the epidermis? And therefore we inserted microRNA 39 from C. elegans, which is specific to the worm, doesn't exist in humans. We transfected fibroblasts with this microRNA, put them into the collagen matrix, generated 3D skin equivalents, separated epidermis from dermis, and then looked by qPCR if we would find this microRNA, this worm-specific microRNA in the epidermis, and indeed we do. So we have an indication that such a crosstalk can happen, at least in organotypic cultures. So if we have such a transfer, then what could be the outcome of this? And if you remember that senescent cells impacted on keratinocyte differentiation in our first experiments, we took the extracellular vesicles from senescent fibroblasts and put them onto keratinocytes, and what we indeed see is that the closure of scratches in keratinocyte monolayer cultures is faster by the presence of extracellular vesicles. On the contrast, if we expose these the keratinocytes for a long time to these extracellular vesicles from senescent cells, we seem to inhibit at least the formation of fully differentiated keratinocytes by seeing that involucrin as a late marker of keratinocyte differentiation is down-regulated after long-term exposure. So we again might have this double-edged sword of senescence, where in the short term it might have beneficial effects, while being chronically exposed to the SASP, you might have problems. Can we get this effect on keratinocytes down to a specific microRNA? One of the microRNA that we found differentially expressed per single vesicle is microRNA 23A3P. So we transfected the keratinocytes with this microRNA alone, and indeed we can phenocopy the effect that we saw with the full extracellular vesicle fraction from senescent cells. Can we find extracellular vesicles now in skin in vivo? For that, we teamed up with Ida Perrotta, a specialist on electron microscopy at the University of Calabria. And indeed, we find in skin cells ex vivo structures, multivesicular body structures with single structures that resemble exosomes inside of the cells. And these structures inside of the cells are positive for different markers of vesicles like DSG101, CD81, or CD63. And actually we see such structures even in the interstitium of the cells, meaning that, yeah, such vesicles are also secreted in vivo, although we don't know the directionality where these vesicles would go to. So we tried to isolate vesicles from human skin by a protocol that we set up, and indeed, yes, we can get vesicles out. But then obviously we are using a lot of enzymes to digest the skin, so it's not... We were not really satisfied at the end with the experiment here. So in that case, we teamed up with Ioneum Research in Graz in Austria, where they developed an open flow microperfusion system where they insert capillary tubes that allow at a certain kilodalton exclusion barrier, the coming in and going out of molecules. It's large enough for extracellular vesicles to cross the barrier, go in these capillaries. So we got hold of some of the samples from probands that look like that. It's considered a minimally invasive technology, though, and but you can also run that in vitro on skin flaps that survive in vitro for a week or 10 days. So we tested if we would find microvesicles in those interstitial fluids that we collect from open flow microperfusion system, and indeed, yes, we get these vesicles. They are in the size, they are similar to what we get out of our in vitro cultures, and also in TEM they look pretty similar. Do we find the microRNAs we were interested in also in these samples? Yes. Yes, we do, and for example, microRNA 23 is one of the highest that we find in there. In these interstitial fluid-derived extracellular vesicles. So, obviously, one of the next steps is to look into young versus elderly subjects, and we are currently trying to get the ethical approval to run such a study. Summarized, I think, or I hope I could convince you that extracellular vesicles and the microRNA cargo are indeed part of the SASP, that we also can dampen this SASP by plant extracts, and that if we don't dampen that, a cargo is functionally transferred in vitro and in vivo, and we can add some messages of extracellular vesicles from senescent cells, which is that they talk about apoptosis to other cells, about differentiation and proliferation. And I think it's now also clear why vampires only bite young girls. It's clearly because they want to dilute their SASP. And with that, I would like to thank all the people in my lab for all the fantastic work, especially Lucy Teletsky-Zaniewicz and Sylvia Weilner, who performed a lot on the extracellular vesicle microRNA studies in my lab. Ingo Lemmermann, who was the genius visualizing the effects of the plant extract here. And you will hear Regina later, so I just say then welcome to you afterwards. And all my fantastic collaborators at Medical University of Vienna, Florian Gruber, who is in the audience, by the way, and Erwin Schachler, collaborators at Chanel Research and Technology, including Julie Latreille, Irina Berlin, Frédéric Morisot, Sandra Forestier, Gaël Gendronneau, and Nicolas Fuzzati. University of Lausanne, with whom we collaborated on the fly work, Ioneum Research, as I laid out, and we are also in contact with Jim Kirkland and Julie Campisi for further developing the senolytic ideas that came up during our studies here. And thank you for your attention, and I'm happy to take your questions.
So thank you very much, Johannes. Cédric? Non, bah alors Jérôme Lamartine.
Well, thank you very much for this very nice presentation. I have plenty of questions that we will discuss later. Just one for beginning. When we speak about microRNA and exosome, the main question is how the message is put into the bottle. So how the microRNA are selected to be inside the microvesicle. So do you have ideas about that? And can we modify this selection of microRNAs? Perhaps, I don't know why. But.
Yeah, absolutely. That's a very good question. So there is a selective process of including specific microRNAs, but then we also have a default mechanism. In all the conditions and stages where we looked at the microRNA of extracellular vesicles from different cell types, from different senescent, non-senescent cells, we see that about 50% of the microRNAs, they just stay the same level. So they seem to go there by default. They seem not to be really selected for to go into the extracellular vesicles, reflect more or less what you would find in terms of levels in the cytoplasm. But then about a fourth of the microRNAs that we find in the vesicles, they are de-riched in the cytoplasm, and another fourth or 25% is enriched in the extracellular vesicles as compared to cytoplasm. So there must be a specific mechanism. There is one study out that shows that there are some sequence-specific signals that lead to the inclusion of some microRNAs, but definitely there is a lot to discover in that regard. Can we manipulate that? We don't yet understand everything so well. We know some steps in the assembly and how the microRNAs get in there. So it is going to be, you can manipulate that. And one thing without knowing really the mechanism how you can manipulate is transfect the cells with microRNAs, and they indeed go into the extracellular vesicles, as we have shown for the worm-specific microRNA, where we don't understand why this is happening. Why does it go into it goes in the endosomes probably, and then that's why and how it goes to the MVPs. But yeah, loads to do.
Thank you.
Cédric Delevoye.
Yes, here. Actually, I have two questions. One is related to more the biogenesis process within the MVB, since these senescent cells secrete more, as far as I understood. I was wondering whether you have a kind of new biogenesis, or it's just a kind of fusion process that would be kind of promoted. And a second question would be more related to the last part, that is, since you are able to collect extracellular vesicles from the fluids, I was wondering whether you can get numbers. I mean, because this is a real challenge now to try to understand whether it's a very dynamic, important process in vivo. So can you have numbers per cells, exosome per cells, whatever?
Mm-hmm. So the first question on was biogenesis of extracellular vesicles. we see that the whole ER and all the secretory pathways are upregulated in senescent cells. That is something that is known. We haven't specifically looked at if the pathway to generate EVs would be up in senescent cells, but that's a very good question. I'll go right back to the lab and look at our expression data there. And the second question was on the quantitation of vesicles in the skin ex vivo. Yeah, so far we didn't do that yet because we had nothing to compare to. That was healthy individuals from a study that Your Name Research did, and where we were able to get some samples. So we cannot yet really compare, young versus old. This is something that we are planning at the moment. In terms of numbers compared to the in vitro situation, we are in a very similar range, and with about the tiny amounts that we get out of the perfusion system of about 100 microliters, we are able to isolate RNA out of that, and we are able to isolate the vesicles similar to, actually even better than from conditioned media. So, yeah, we are in the beginning of quantitating that and having the comparison, hopefully in a couple of months, which then at the end will be a couple of years.
Thank you very much, Johannes. So right now we will have the poster session and the coffee break, and we will come back here after 30 minutes.